Real quick and talk about what enzymes are. The most important thing that you know is that enzymes are biological catalysts. So when we're talking about biological, what we mean is that they are made out of macromolecules. They are made out of molecules that are made in the cells. And in this case, the macromolecule that we are talking about is a protein. So we're going to spend a little bit of time going over protein structure because it's going to be really important for understanding what enzymes do. The monomer of proteins are the amino acids, which, as we remember, is made out of carbon, hydrogen, oxygen, nitrogen, and this R group, which can include sulfur, but the R group is really that variable group that can be different depending on what the amino acid is. And so when two amino acids come together, they're going to begin to form like this strand, and eventually what we end up with is a whole bunch of amino acids that are strung together, and each one has its own little R group. And so eventually this big old strand is produced that can be dozens or hundreds even of amino acids long. Those R groups are going to be really important because it's going to be the R groups that dictate how the protein folds. The R groups are going to sometimes have positive areas or negative areas. Sometimes they can even covalently bond to each other, and they're going to do this with other R groups that aren't necessarily right next to them. For example, maybe one of the first R groups in the chain is going to interact with one of the last R groups in the chain. And so what you end up with is this very complex folding pattern that all proteins rely on. So their three-dimensional protein folding is going to give it a shape. And so here's an example. This is hemoglobin here. And these tiny little things that look like wire, those are the amino acids, and what dictates how they fold is how the R groups interact with one another. And we can look at this as a space filling model. Looks a little more like what you see on the right. So big picture here to remember is that proteins have these complex folding patterns that dictate shape, and it is that shape that determines the function. So when you think proteins, think about shape or three-dimensional like folding patterns, because that is what is going to be the most important. Okay, so we know that enzymes are biological catalysts. So now we need to really dive into what a catalyst is. And so any catalyst is going to be a molecule that does something very important, namely it reduces activation energy of a chemical reaction. So enzymes are therefore going to reduce activation energy. And what do we mean by this? Well, if we start off with a chemical reaction, you need to remember that the reactants on the left are only going to be able to form products, shown here on the right, if those reactants become destabilized. The methane, the CH4, and the two oxygen molecules being shown aren't just going to willy-nilly decide to break apart because they're in a stable configuration. So the very first thing that you need to have happen in any reaction is that the reactants have to break apart, that bonds have to break. And the only way that you're going to break bonds is by adding energy. So that energy is called the activation energy. Just as a reminder then, we have the reactants being shown on the left, the products on the right, and it's that intermediate stage where energy has been absorbed that we're talking about here, right? That little hump in the middle of the graph is our activation energy. And we can look at chemical reactions that have more energy at the end than when we began. That means that more energy overall has been absorbed by the reaction, and this is an endergonic reaction, usually the kind that are associated with the building of molecules, such as anabolic reactions. Or we can have a catabolic reaction, or something where complex molecules are broken down. And when that happens, you have the energy that is being released. Therefore, what you have at the beginning is more energy in chemical bonds as compared to at the end. But regardless, it doesn't matter what kind of reaction we're talking about. It's that hump, that piece right there, the energy needed to start the reaction, that is the activation energy. And again, enzymes are biological catalysts. Catalysts are molecules that reduce activation energy. In other words, they're going to make it so that the reaction can happen without having to add quite as much energy into the reaction. Now notice, you still have the same amount of energy in the reactants and in the products. That doesn't change. All you're changing is the amount of energy that needed to be added to get you over that hump so that the reaction could get started. And the way that enzymes are going to do this is, again, remember that proteins are three-dimensional, and it's their shape that determines the function. In this case, the enzyme has a shape that perfectly fits what is shown here as maltose, which is a disaccharide, a sugar that has two monosaccharides combined together. And you'll notice that the maltose perfectly fits into the enzyme. The part of the enzyme where this binds right here is called the active site. And then the thing that it binds to, the reactant really, is called the substrate. So the substrate in this case is maltose, and the enzyme's active site is going to bind to that substrate. And when that happens, it's going to change the shape of the enzyme ever so slightly, and in such a way that it causes stress to the bond. So instead of having to add a ton of energy to break that bond's a little bit more stressed. It's a little bit more likely to break on its own, so not as much energy is needed to break it. I like to think of enzymes as little chemical factories, right? They're taking whatever the reactants are, putting them together in such a way that it increases the likelihood that bonds will break and then be able to reform. So in this case, we've broken the bond. The maltose pieces can now react, and in this case, they're going to form glucose, which are your monosaccharides that are most important in the body. So nifty begifty. So again, enzymes are biological catalysts. They're reducing the activation energy in a reaction. And why is that so important? Well, we have chemical reactions going on all the time in our bodies. And if we needed enough activation energy to start them without enzymes, you'd basically be lighting yourself on fire. So they're allowing chemical reactions to happen at biological temperatures. We don't need to light ourselves on fire. They can happen a little bit more readily and at a temperature that is not going to destroy your cells. And as I said, they're happening way more rapidly. It's a lot easier to overcome a small little hump of activation energy than a huge one. So the reactions can happen super quick. We're talking millions of times per second. And so just kind of to wrap up here, some important information about enzymes. They're specific. We showed earlier that the active site and the substrate have to be able to bind, and that is true. Most enzymes are only going to be able to catalyze one reaction, and usually that enzyme is going to be named based off of what the reaction is that's being catalyzed. So for example, earlier we saw maltose, which is being broken down. The enzyme in that case would be called maltase. So most enzymes end in ASE, and that's going to be kind of your tell to know that it's an enzyme. Enzymes are going to be affected by their environment. If you make sure to read through chapter 2.4, there's a lot of different conditions that will affect how rapidly an enzyme works or completely prevent it from working, including temperature and pH, as well as a number of other factors. And enzymes are proteins, and they're coded for the DNA, which means that depending on what your DNA is, you're going to have different sets of enzymes. And so that means that not all organisms are going to make the same enzymes. Even within a species, you might have someone that has a version of an enzyme that doesn't function properly. And that's a basis for a lot of different diseases. And not only that, but the cells within a multicellular organism aren't always going to make the same enzymes either. So there's some that are going to be really, really important that all of the cells in your body are going to make, but there's other ones that are more specialized for particular types of cells. So I know that was a lot of information. I hope it at least supports some of what you take the time to read in chapter 2.4 so that you are understanding this concept a wee bit better for when you come back on Thursday. And with that in mind, I hope you enjoy your trip to New York and catch you on the flip side.